Welcome viewers. Today we are going to discuss on antimicrobial resistance. Antimicrobial resistance means the ability of the microbes to resist the action of antimicrobial agents. This antimicrobial resistance can be shown by bacteria, viruses, fungi and parasites as well. This ability may be their inherent ability to restrict the effect of the drug, or it may be the ability that they have acquired in the due course of their growth in the environment. So, antimicrobial resistance is a matter of global concern in the 21st century. The reason behind the drugs are becoming ineffective and antimicrobial resistant strains are growing like anything, as a result of which the treatment is getting prolonged and we need to hunt for newer varieties of drugs to combat the pathogens. That's why it has become need of the hour to have a clear-cut idea about the modes of development of antimicrobial resistance. Before actually entering into the study of methodologies of developing antimicrobial resistance, we would like to focus on the factors that are responsible for making the drug an effective for use. So, the first thing about an antimicrobial drug is that the drug should be able to get access onto the pathogen where it has to execute its killer action. As we have seen that there are certain drugs like penicillin G which cannot be taken orally. The reason behind is that penicillin G is very much sensitive towards the gastric pH of the stomach.
Ma'am, we have heard about parenteral routes of drug administration. Okay. Ma'am, what is it actually?
Actually, many a times we cannot administer the drug orally. So, we use some other pathways for administration of the drug. Say for example, intravenously or intramuscularly the drugs are injected. So, this ways of injecting drugs other than the oral route is called parenteral route of administration. As for example, we have seen that drugs like gentamicin or aminoglycosides, they are administered intravenously or intramuscularly. The second point that comes in our mind is that the drug must be, or the organism must be susceptible to the drug. Means we are administering the drug, but that drug should be able to impose toxic effects on the host. As we have seen in case of biofilms, many a times the organisms or microbes residing inside the biofilms are showing immense resistance towards certain class of antimicrobials.
Ma'am, why so?
Basically, the entry of the drug inside the biofilms are sometimes restricted, number one reason. And number two, when they are residing inside the biofilms, there are many microbes residing together, and that actually slows down their growth because the nutrients are getting depleted. So, they replicate at a slower rate, and their metabolic steps are being slowed down. And as we all know that most of the drugs are antimicrobials that target the replicating cells and they target the metabolic pathway of the cells. That's why if the bacteria is becoming slow growing, then obviously the effectiveness of the drug also gets depleted. And the third thing that is very important, that MIC value or minimal inhibitory concentration value of the pathogen should be exceeded by the chemotherapeutic agent. And the concentration that reaches the target site or that reaches the pathogen site actually depends on the amount of drug that you have administered, number one, and number two, the route through which the drug is being administered, and number three, the speed with which the drug is uptaken, and number four, the rate at which the drug is cleared or eliminated from the body. As we have seen that every year almost 4,40,000 patients are showing signs of multiple drug resistant tuberculosis. This rate is really alarming, and almost at least 1,50,000 patients are dying annually because of MDR-TB. Antimicrobial resistance may develop because of two different causes. Number one cause is because of spontaneous mutations, and number 2 is acquisition of some genes from other bacterial species or from environmental samples. We have seen that certain antimicrobial agents might be targeting certain ribosomal proteins as aminoglycosidic, antibiotic, streptomycin. So here we can see that if because of certain spontaneous mutations which are occurring every now and then, and the frequency of mutation is say out of 10 to the power of 8 cells, one cell gets mutated, and because of this mutation, if there is alteration in the gene that codes for the protein which is the binding site of streptomycin, then it will lead to development of resistance against streptomycin. Right? But now we have developed certain modified drugs that binds to more than one target sites onto the same ribosome. Under such circumstances, we have seen that the development of resistance by means of spontaneous mutation is very less. The second method of developing resistance is called acquired by means of entry of genes via transformation, transduction or conjugation. These are the basic 3 methods by which a bacteria can procure foreign genes. And once these foreign genes get entry inside the bacterial cells, they may code for certain important enzymes that can actually inactivate the drug, or sometimes they modify the drug in such a way that the drug can no longer execute its inhibitory role. So, acquired resistance is an immense area which we will be discussing in details now. Before going into it, again we can say that bacteria or antimicrobes or microbes can show resistance in 2 ways. One is called innate resistance and second is called acquired resistance. Innate resistance means that bacteria, by virtue of its characteristics or cell wall features or cell membrane characteristics, can restrict the entry of certain drugs or can be resistant towards certain antimicrobial agents. Under such circumstances, they are called as innate resistance. As for example, we can cite the example of mycoplasmas. We all know that penicillins or cephalosporins are cell wall biosynthesis inhibitors. But mycoplasma shows resistance towards penicillins because mycoplasmas are devoid of cell walls. In a similar way, we have seen that there are certain gram-negative bacteria that show resistance against penicillin G. The reason behind is the size of penicillin G is so large that it cannot enter inside the gram-negative bacteria. Third example, as we have seen, the case of tetracyclines, wherein certain bacteria have got certain genes that code for certain pumps that efflux out or push out the drug from the cell, as a result of which the bacteria will start show resistance against tetracyclines. We have seen also that there are certain strains of Staphylococcus that have got the inbuilt gene called beta-lactamase within their chromosomes. Because of the existence of that gene that codes for beta-lactamase enzyme that cleaves the beta-lactam ring of penicillins, this bacteria are from the very beginning showing resistance towards beta-lactam ring containing antibiotics like penicillin or cephalosporin. And we have seen also that under certain cases, some bacteria have got inbuilt efflux pumps. These inbuilt efflux pumps actually pushes off the drug when it gets entry inside the bacterial cell. So, in all we can say that there are various methods whereby bacteria shows resistance in an innate way. That is called innate resistance, which is shown because of its own characteristics. In our next part, we will be discussing about the acquired resistance, whereby actually the bacteria was sensitive towards that drug at some course of time, but due to our misuse of drug or some other properties or some other means, it has become resistant towards a drug. Drug inactivation mechanisms are mainly shown by plasmids called R plasmids or resistance plasmids, which are procured by the host cells or by the bacteria by means of transformation, transduction or conjugation. Generally, these R plasmids code for certain enzymes that actually acetylate, phosphorylate or modify the drug and thereby convert it into an inactive form. As for example, chloramphenicol. Chloramphenicol is modified by a gene called CAT gene, wherein this CAT gene codes for an enzyme called chloramphenicol acetyl transferase that acetylates chloramphenicol molecule, and acetylated chloramphenicol is an inactive drug. So, this is the way how the drugs can be inactivated by the enzymes which are being coded by the plasmids, and these plasmids are procured by the bacteria by the three mechanisms as I discussed. So, the drug inactivation is a very important methodology whereby a bacteria develops resistance against any antimicrobial drug. As we have seen, the another important example is the beta-lactamases. This enzyme is coded by a gene called BLA gene, which is inactivating or cleaving the beta-lactam ring, which is present in penicillin and cephalosporin type of antibiotics. The second important method of developing acquired resistance is decreasing the drug permeability or by increasing the drug elimination. Decreasing drug permeability means the entry of the drug is restricted. As we have discussed before, that there are many gram-negative bacteria that are normally showing resistance towards penicillins, right? Similarly, many organisms are showing resistance against tetracyclines because these organisms develop certain efflux pumps. As in case of aminoglycosides also, we have seen that multiple drug resistance pumps are there that are responsible for pushing out the drug from the bacteria. The third important criteria is altering the drug receptors. It means the drugs get entry inside the bacteria taking help of certain receptor proteins. So, if these receptors are being altered, then we will see that the pathogen will start show resistance against the antimicrobial agent. As we have got the examples of penicillin resistance Streptococcus pneumoniae, then we have got methicillin resistance Staph aureus, we have got vancomycin resistant enterococci. All these resistance are developing because of alteration in the drug binding site. The fourth methodology wherein we can see the development of acquired resistance is that the pathogen starts making certain alternative metabolic pathways to overrule the toxic effect of the antimicrobial agent. As for example, we have studied that sulfa drugs such as trimethoprim or sulfamethoxazole are targeted to inhibit the folic acid biosynthesis pathway. So, there are bacterial strains that have bypassed this pathway and they have developed methodologies to procure folic acid from the environment, so that they no longer depend on the folic acid biosynthesis pathway which is being restricted by the sulfa drugs. This is one of the methodologies whereby alteration in the metabolic pathway has helped the pathogen to develop resistance against certain antimicrobials. Now we are going to discuss the methods to slow down the emergence of antimicrobial resistance. as we have discussed in our lecture now, that it is growing in an alarming rate and it has become a real threat for the physicians and for the patients too. It is a combined responsibility of the entire society to take necessary steps to slow down the emergence of antimicrobial resistance. So, the people who are mostly responsible to slow down the emergence of antimicrobial resistance are the physicians, the health workers, the common people, and the educated society. So, let us start with the responsibility of the physicians or the medical practitioners. The physicians and medical practitioners should be very much careful while prescribing such antimicrobial drugs. Before prescribing any antimicrobial drug, they should do proper tests to check the presence of a particular pathogen. Accordingly, they should decide that what antimicrobial drug should be administered. There shouldn't be any misuse of antimicrobial drugs, as misuse of antimicrobial drugs actually promotes development of antimicrobial resistance. They should make the common people or the patients aware of importance of this phenomena, that how misuse of drugs may lead to antimicrobial resistance. Patients do have a vital role to play to slow down the emergence of antimicrobial resistance, as many of the patients or we have got the tendency of quitting off taking the entire course of antimicrobial drugs. And secondly, many a times some drugs are being administered to be taken thrice a day and we are skipping one of the doses. That is to be prevented meticulously. The reason behind is the antimicrobial drugs therapy is designed in such a way that their concentration within the blood should remain at a particular concentration, and if we skip any of the dosages, then gradually the net effective concentration of the drug within the blood gets decreased, and as a result of which the least sensitive bacteria will start growing, and this will gradually lead to emergence of drug resistance. This overuse of antimicrobial drugs has led to the emergence of severe antimicrobial resistance. So, we have seen that the countries have really become concerned to put a check over the development of antimicrobial resistance. So, one of the best methods to put a check over the antimicrobial drug resistance development is to stop the use or the sale of antimicrobial drugs in prescription independent manner. As we have seen that in developing countries, the drugs are being administered or being sold, though they are not being prescribed by the physicians. So, this practice is to be stopped or banned completely. As the drugs are sold without the prescriptions, they are being prescribed by some illiterate people, and people consume the drugs even though without knowing the proper usage of the drugs. There comes the misuse of drugs. So, the first point that is to be followed very meticulously is to ban the sale of drugs without any medical prescription. The second important concern is use of this antimicrobial drugs in animal feeds to avoid the animals to get infected. So, they are being given this antimicrobial drugs, as a result of which we are getting good quality flesh or good quality animal products at a very low cost. But in a way we are promoting the development of antimicrobial resistance. It has been seen that various resistant strains of Salmonella are raising because of the animals being treated with the drugs. So, this type of drugs are to be completely banned in the animal feed. We have considered what is antimicrobial resistance, how antimicrobial resistance developed. We have seen it may develop due to spontaneous mutations or it may be a acquired phenomena. Then we have also seen that the pathogen may acquire the resistance gene via the plasmids or transposons, and the way of entry may be transformation, transduction or conjugation. The bacteria may show innate resistance. Innate resistance is precisely due to its own cell wall, cell membrane characteristics, that it restricts the entry of certain antimicrobial drugs, or it may have certain efflux pumps. We have also discussed the various ways of developing acquired resistance, wherein we have discussed about the drug resistant plasmids called R plasmids that may code for genes that nullifies the drug or modifies the drug and inactivates the drug. We have also discussed about MDR pumps and the development of MDR pumps because of spontaneous mutation sometimes, and because of some other ways we have seen that the rate of mutation or the rate of emergence of antimicrobial resistance is becoming a global threat in these days. We have also discussed the different roles to be played by the physicians and health workers to restrict the emergence of antimicrobial resistance. And we have also seen that the patients should be well aware while taking any antimicrobial drug.